says to make sure everyone has our tools.
Yeah, I think today would have been a terrible day.
I'm going to make sure that
We hit the window perfectly.
Our tools is,
And then we went
microphone.
to
most people should have it,
a place with a lot of apple cider.
but chain yard.
Chain Yard. Yeah.
Is this the one?
Hello? Can you hear me? It's good.
Talk that if you like.
One more button, maybe. Go.
Jake, does the microphone so I can introduce you? Sorry. You can use chest. I will take it though.
All right, is everybody just about ready to get started again? All right, everybody's now ready to get started. I hope you all had some delicious snacks. I'm really happy to present our next speaker here today. We have Jake Brownscombe, who's a postdoctoral fellow with the Banting Scholarship Program here at Dalhousie University with the Ocean Tracking Network. Jake and I have worked together for many years. First met in a crazy trip to Brazil in 2014 where we did some research on snook together in the Cananea estuary. And since then, Jake and I have worked very closely for the better part of the decade on various questions related to fish tracking and accelerometry and scientific questions. Jake did his PhD at Carleton University with Steve Cook and moved down to Florida to study permit, and not that kind of permit, but the fish. So he's doing a lot of work tracking these fish down in Florida, and Jake's done a lot of thinking about some of the fundamentals of telemetry and science and how different approaches work, and really thinking critically about the way that we're implementing a lot of the tools that we have. So this is a really important presentation that's going to touch on some fundamentals of array design and testing that have huge implications for the way that we're conducting our research. So appreciate you coming, Jake. Thanks so much, and I'm really looking forward to your presentation.
Robert. Assume that clapping was for Robert. Haven't done anything yet. Hello, everybody. Thanks for coming out. Huge thanks to OTN for putting this on and for having me supporting my travel out here. Special thanks to Rob and Kim for putting this together. It's a fantastic workshop and a really great group of people. So I feel very fortunate to be here and spend time with all of you. So I'm going to talk today. I'm going to talk to you for a little while, and then I've got some R code. And it's all sort of based around this paper that we came out with towards the latter half of last year, where we aim to develop a more tractable and easy to implement practical approach to measuring and accounting for variation in the performance of telemetry systems, to help to avoid the performance of the system, which is changing over space and time, from influencing the patterns that we're trying to pull out of the actual ecology of the animals. And so before I dive into that, I want to talk really quickly about collaborative data analysis too. It's something that isn't really being covered a whole lot in this workshop, but from my perspective, it seems like many of us analyze data where we have a single individual working with all of the data in an R script that goes into a manuscript, and then all of the co-authors review the manuscript, but very few co-authors tend to review the actual analysis in the R code. And there's huge potential for errors during that, all of that processing of the data. And so what I've started to do is go over to using GitHub and using that to remotely collaborate with my collaborators. It's a really fantastic forum for doing it because it interfaces directly with R. You can push and pull the R code and data in and out of GitHub and work with your collaborators remotely for that. And there's also version control. So any single collaborator can't mess up the data set too badly. And so If this is something that you guys are interested in, you wanted to get set up with it, it's pretty easy to do. Just come talk to me afterwards. And John knows a lot about it. People at OTN know a lot about how to use GitHub and how to collaborate. So it's really quite handy. It's something I'd encourage. So back to the detection efficiency stuff. This is not the most glamorous part of telemetry, but I think something very important and something that we often ignore or don't know what to do about. And so last year we also came out with this paper where we outlined what we consider to be the most important considerations for, I'm going to go out of camera a lot, for telemetry analysis. And Nathan did a fantastic job of covering a lot of these basics yesterday. The part that we're going to focus on today is in accounting for system performance. We know, there's lots of discussion of this yesterday, that for various reasons, all of the transmissions that get sent from a tagged animal in proximity to a receiver may not actually get fully decoded and measured and saved by that receiver for various reasons. A big one is physical obstructions, and these can vary a lot over time. So you might work around places with seasonal vegetation, or changes in you might have ice cover for part of the year. So things that are changing the ability in line of sight from between the tag and the animal might change over space and time. You also get code collisions. So the more animals that are in your system in proximity to the receiver, the actual less detectable other animals are. And this is the case for VEMCO equipment anyway, because it's operating all on the same frequency. There's other systems that get around this, of course. And then, of course, environmental noise is a huge one. The systems I work in tropical, snapping shrimp and fish calls are a huge one. You also get influences of abiotic factors like wind. Wind is a massive one, and currents. And then you have anthropogenic sources, things like boats driving by. These are all impacting the performance of receivers over space and time. This is just an example. The VEMCO TX receivers now record some mean value of noise in millivolts. And so we have our sort of extended team has some receivers on the Florida Reef Tract. And so this is just an example of how the noise values vary seasonally. We get these really interesting peaks in the summer, which I think are probably associated with biological activity, whether it's shrimp or fish occupying this site. And so this has potentially huge impact on our ability to measure whether fish are there or not. You have almost twice the amount of noise in the summer as the winter. So if you get twice as many detections in the winter than the summer, are they really using it twice as often in the winter? Or is it just the noise that's influencing it? To add to this complication, we can't seem to use noise directly to account for detection efficiency variability. So this is the relationship between that noise metric on the x-axis and the number of detections that we're getting from stationary transmitters in the system. And there's actually quite a poor relationship between it in this case. I don't know why. I was talking with a few different people about this yesterday. It's clearly there's something else going on, at least in this system, that means detection efficiency varies independent of noise for other reasons. It could potentially be wind or how smooth the surface of the water is that's reflecting signals. I don't know for certain. Or perhaps this noise metric isn't comprehensive enough in the current VEMCO metrics to measure the actual impact on detection efficiency. The reason why we need to control for it is really well illustrated by this paper led by Nick Payne in 2010. I encourage you all to check it out if you have not yet. It's a decade old now. But they were tracking cuttlefish and looking at their use of nearshore areas. And they decided to put out some reference tags in their system. And what they found was that the raw detections would have suggested they were using nearshore areas the most during the day. But in fact, when they accounted for system performance in detecting the reference tags, they were actually more using nearshore areas at night. So this is an example where you could completely misinterpret the dial patterns of space use in an animal because of the system performance. The question still remains, how do we measure system performance over space and time? That Payne example, they threw out some reference tags in their system, but there wasn't a standardized way to measure performance of all the receivers over both space and time. How many reference tags do you put out? Where do you put them? And how does that actually derive some kind of measure that you can apply to your detections to correct them? The typical approach to measuring detection range, detection efficiency is well illustrated with this example. where many studies in the past have gone out and put reference tags that are just in stationary positions at set distances from the receiver, and then they're constantly pinging away, and you can measure detection efficiency over time. And it gives you plots like this. And so you can see there's there's a range, there's a huge variability in the detection efficiency over space and time in this system, as we see across all telemetry systems. And despite knowing that this is important, Steve Kessel led a study in 2014 that assessed how well telemetry studies were accounting for system performance in their analyses. And basically what they found is that the vast majority of them scored very poorly in this. So this is something that as a community we don't seem to be doing a very good job of to date. And so our thought was if we could come up with something that's a little more tractable and simple, logistically practical to use, people might start using it more. And so we came out with this paper last year in Methods in Ecology and Evolution that represents, it's a flexible framework, and you could do the bare minimum, or you could do more if you have more time, if you have more resources, financial and time. But this represents what we thought would be sort of the bare minimum example of how you might account for performance over space and time. And I would like to point out that it may not necessarily be always important to do this. For example, if you're just interested to know if your animals are emigrating from an area and how many of them do that, you might care about detection range in designing that gate of receivers so you know how far to space them, but you might not necessarily be too concerned with correcting for detection efficiency over space and time in every situation. But if you are interested in spatial and temporal patterns of animal space use, this is clearly a relevant thing. So this is basically the framework that we came up with. So you take your acoustic receiver array, in yellow, those receivers, and you pick a set of sentinel sites. These are going to be sites where you're going to do some range testing and monitor detection efficiency over time. So you should select those sites that represent, it's very important that you select sites that represent the entire range of conditions across your system, because you're going to develop a model based on those sites and then make predictions throughout your entire array about what detection efficiency is like. And so you need to capture all those conditions, all the different substrate types, different water depths, potentially different fetches and angles towards the wind, for example. So this is our array in the Florida Keys. This is Key West right here, big party town. And we have all our receivers out here. We're tracking a bunch of different coastal fish species. And we selected these 9 sentinel sites across our array that in some part we tried to capture all the different conditions, but was also logistically feasible for us to go out and do a lot of this stuff. At each one of those sites, the very first thing that we did was conducted a range testing protocol. And we used a very, very basic one where we went out, we tried to find the calmest, most ideal conditions, so high tide, very low wind. And we just took a range testing transmitter that's pinging every 10 seconds and put it off the side of the boat for a couple of minutes at a whole range of distances from these sentinel receivers. And what you get from that is this. You get a relationship between distance from receiver and detection efficiency. You know how often the tag is pinging, you know how many of those pings got picked up by the receiver once you've downloaded it, and you can plot out this relationship. I then fit those relationships with a third order polynomial with the set x or y-intercept at 100. And using that model, I'm able to predict the maximum range, which I perceived as being where detection efficiency is 5%, as well as the midpoint, where detection efficiency is 50%. And the reason that I care about the midpoint is because we're going to place a reference transmitter at each one of these sentinel sites that's pinging away throughout our entire study at that midpoint. And by doing that, the idea is that we're enabling each site to have the same freedom of variance around the mean. We're aiming for that 50% mark. I did not do a very fantastic job of achieving that here, which could be for a variety of reasons. Probably more extensive range testing at the start would have given me a more informed, better informed placement for the reference tag. But I still have some measure of how the system is performing over time, variance around that mean. Yeah, Rob?
What's the freedom of variance? Standard error or?
Here?
How do you define freedom of variance? Can you repeat the question?
How do you define freedom of variance? So what I mean is that if you had a reference transmitter out where the majority of the detection efficiencies are close to 100. That reference transmitter can vary a lot downward, but not upward. Do you know what I mean? Yeah. So they all have the same freedom of variance, the same potential to go up and down and vary over time. Increase in detection efficiency, decrease in detection efficiency. Does that make sense?
I think so. But essentially, it's just setting a ceiling of 100%.
If you put the tag too close to the receiver, it's just going to hear it all the time. And you're not going to capture that variability that's going on. You put it too far away, it's not going to hear it enough. And you're just going to get a lot of zeros when there's like other things going on in that curve. So you're trying to capture the slope of this curve. So you can get this variance that's going on in here. Because if you're out here too far, you're not going to capture it. If you're here too far, you're not going to capture it, is the idea. Not thrilled with it, but it sort of worked out. So the next thing that we need to do is we need to combine that initial range testing of the maximum range with the information on how detection efficiency is varying over time to derive some sort of standardized detection range correction factor. Some equations, I'm not going to dive too much into these, but essentially at every study hour or any period that you're interested in, you can calculate the detection efficiency variance, so the detection efficiency at the time minus the mean at that sentinel site. And then we came up with this equation where essentially we're combining that detection efficiency variance metric with the detection range, the maximum range. You then have this DRC. This is the detection range correction factor, the standardized factor that's varying over time and space that you can use to correct your telemetry data. And you can do that in a whole variety of ways. The simplest way would be simply using this equation, divide it by its mean, and then divide the number of detections by that value. That's going to give you some standardized measure of the number of detections that's accounting for variation in detection range. Yeah?
What was the ping rate of those sentinel tags?
The reference tags are pinging, I think 270 to 330 second range.
Okay.
Yeah, and understanding those ranges, how often you're expecting your detections is very important. And with VEMCO tags, you also need to know that there's a ping train interval. And so if your transmitter is pinging every 7 seconds, it's going to take 3 seconds to actual ping. And so your delay is actually 10 seconds. You would expect it every 10. That's a really simple example of how you could correct number of detections. I think this DRC, this detection range correction factor, could be integrated in all sorts of analyses. And I'll talk about some examples later. So in this paper, we applied it to a hypothetical data set just to see how it would perform. So we generated a hypothetical data set where detection range varied randomly between 50 and 500 meters. Detection efficiency variance went up and down from negative 50 to 50%. And we just simulated 0 to 10 detections per hour. And so you can see how this detection range correction factor is derived. It's coming from that maximum range is the main driver of it, and then detection efficiency variance over time changes it up and down. And so this is a unitless measure, but it could be considered very similar to meters. You apply this detection range correction factor to the number of detections with that equation I showed you before, just as a really simple example. And you can see in some cases, hypothetically, you can get major differences between the raw and the actual detection values, up to over 1,000% difference. So hypothetically, detection performance and the performance measured and derived with this DRC value could theoretically impact your number of detections quite substantially. The next important thing you need to do is model that DRC value over space and time so that you have a model that you can predict into the rest of your system. Because we don't have the time or resources to monitor detection efficiency at every single one of our receiver sites, this enables us to take that data from the Sentinel sites and predict into the entire system. I did that with random forest. This is a type of machine learning algorithm. Random forest in particular and a number of different machine learning algorithms are really, really handy models in ecology. Something I think is underutilized because they can handle a lot of different types of data that typical statistics cannot. So if you're going to fit a generalized linear model, there's a lot of assumptions about the data, the distributions. You can't include correlated predictors. whereas random forest can handle all of those things. It can handle missing data, it can handle small data sets, big data sets, outliers. It's insensitive to monotonic transformations, so you don't really need to worry about log transforming your data, for example. It doesn't make any assumptions about the distribution of the data. And it can, in some cases, be very accurate at predicting. So this is, if you're interested in these, I do have a machine learning workshop applying machine learning stuff in ecology. You can come talk to me about it afterwards. I think it's on my GitHub page, actually. There's a bunch of stuff on my GitHub page. If you go there, my username's Jake Brownscombe. This is also a fantastic paper. Outlines random forest and ecology. And then there's another type of model that's kind of similar called boosted regression trees. It's a little bit of a different approach, but they also function very well on ecological data. And this paper is fantastic. It gives you all the R code to fit these models as well. So I fit a random forest to the sentinel tag data to model that detection range correction factor, that DRC, over space and time. And I'm integrating the predictors into this model that I'm interested in the actual fish data. So if I want to know how fish are using my different habitats across day and night, I'm going to include habitat and day and night in my model. And that way I can make predictions at the same ecological time scale as my detections. So I fit this model, this random forest model. I'm not going to get too much into how it actually does it. But these are the predictor importance scores. So you have these site-specific factors that are very important. Things like benthos type, water depth, habitat type, and rugosity were the most important predictors. And then these temporally changing ones had an effect, but less of one. These are less important. So tide height, tide state, and dial period. The model is able to describe 76% of the variance in the data, which is not fantastic, but not terrible. You could probably put a lot more effort into the modeling and improve this a bit, but it's not doing terrible. So now I have this model that can predict DRC over space and time. And so this is just an example. Key West again here is on your right side. And so I'm able to predict at a bunch of receiver locations that we didn't even measure detection range at, just based on their characteristics and the time period at the time. And so this is just showing that DRC value, the size of the circle, not exactly meters, but could be considered sort of analogous to that. And then it's just colored by habitat type. So you can see, like, for example, this really rugose tiny channel just hears very poorly compared to this more open, flat sandbank, which makes a lot of sense. So if you're getting 5 times as many detections here as here, you got to think a little bit about what that means. Right? You're sampling a much larger area in that habitat type. So you now have this model, you can make predictions, and you've got this correction factor that can be used in a variety of ways to integrate into your modeling of animal ecology. In this paper, I just did a really simple example using that equation that I showed, where I just divided the DRC by the mean and then divided the number of detections by that value. Really simple application, and then you see the patterns in the raw data in the red compared to the corrected data in the blue. And we see some differences. Particularly, they seem to be using the channels, which don't hear very well, particularly during the day, apparently, because you see the model is now upping its proportional use of that habitat type in the day. I did this with some regret in retrospect, because I don't think that this is really the best application of this correction factor to telemetry data. It's a really simple one, and I'm surprised that the reviewers let me away with this. But I think realistically, you're probably more interested in using this DRC value in a more complex model. Something like a generalized linear model of presence-absence over space and time. You could include that DRC value as a random or a fixed effect in those models, for example. You could integrate it into occupancy modeling frameworks, hidden Markov or state space models. It could easily be integrated into those, or the interpolation methods like the COAs that people commonly use, centers of activity, which we just calculated in Ryan's workshop. A couple of good papers that describe methods that would easily uptake this DRC value. This paper led by Joseph Allos, a state space modeling framework that integrates receiver detection efficiency error. That DRC value could go straight into a state space model like this. the interpolation methods, those COAs. This is another great example by Megan Winton. Came out a couple of years ago, where she integrates the variation in receiver detection efficiency or receiver performance in those COAs. Again, DRC could go straight into this model, for example. Is that straightforward to everybody, sort of? Makes some sense? Cool. Yeah, again, all the code associated with that methods in ecology and evolution paper is it's online in my GitHub. All the data is available as well. And so we'll go over, John, where's John? And pop into R here for a minute. Yeah.
If you had, when you have your Sentinel, are they always Sentinel receivers, or can you have a Sentinel site composed of a number of receivers? And still get a measure. Some of the places where Danielle and I work, we know it's so bad. It's the Bay of Fundy tides. It's turbid, muddy.
So you got a bunch of
what we just like, well, how many receivers we have? It's a gate kind of thing.
Right.
We're putting as many as we can, and it's still bad. So have you ever? Can it be applied like that, or would you need to calculate it for each receiver?
Yeah, you could put a reference tag out in proximity to a group of receivers. The thing I like about that midpoint approach is you have this standardized measure that you can apply at a population level of your receivers. Whereas if you have a reference tag at a bunch of different distances from receivers, it's not a standardized measure anymore. It's hard to know how to combine them. What does 50% detection efficiency at 100 meters versus 75% detection efficiency at 200 meters at a different receiver? How do those two jive together and produce some kind of standardized correction factor? Yeah, maybe something we could talk about afterwards. And I would like to highlight the fact that this framework that we came up with is theoretically pretty flexible. So that was sort of like the bare minimum, what we thought you could do. But if you have more time and resources, you could put 3 or 4 or 5 reference transmitters out at each of your Sentinel sites. You do have to think about the fact that we use 9, so we built 9 extra moorings, bought 9 reference transmitters. If you wanted to use 3 at each site, now you're talking about 27 moorings and 27 reference transmitters. And you got to think a lot about where you're going to put those 3 reference tags and how you're going to get a standardized measure across your whole system that can be combined into one standardized correction factor. So all the code associated with this method is available online through my GitHub, and you should be able to all access it through the OTN GitHub page. Through Kim's GitHub page.
For today, the one on Kim's.
Yeah. Yeah, I think you could aggregate your data at whatever time scale you're interested in ecologically. So if you're going to aggregate your data by day and night across seasons, that's the number you're interested in terms of number of detections with your animals. You could also calculate the mean detection, the mean DRC value across those same levels and use that as a correction factor. And the nice thing about using the model to predict DRC over space and time is that if you use the detection efficiency of a reference tag at a certain site as a direct measure, any kind of site-specific impacts on that detection efficiency might get extrapolated across the whole system. Like if you had a boat go by one site while it was measuring it, and it's just something that's like a little, doesn't represent like the overall pattern in the system, if that makes sense. So you guys have this sheet open? Everyone able to access it? Got the colored things going, the sheets? So there's a few packages you're going to have to call. If you want to just go ahead and call those, you might have them up already. Most of these have been used already. ggplot, ggmap, gganimate, reshape. It's just for manipulating data frames. All that stuff could actually be done in dplyr if you wanted. And then we're going to use a random forest at some point. So there's kind of 3 different data sets here. You've got the range that initial range testing data with that range testing tag. You've got the detection efficiency from those reference tags at each Sentinel site over time throughout our study. I think we published 2 months of data with this. And then you have our detections of permit in the Florida Keys. So we'll bring in our reference or range testing data. We can have a look at it. This data has had some processing done to it already. So I've gone in, I think probably just in Excel, and you know, this number of detections per hour is something that you're going to have to pull out of VRL, right? And actually count up the number of detections that you get, and then calculate based on your expected detection rate what that detection efficiency is. So here I've aggregated some information about the site, the distance of the reference transmitter from the receiver, the water depth, the tide at the time, and the number of detections we got, detection efficiency based on the number of detections, and that same value as just expressed as a proportion. So if we just quickly plot it by habitat type and receiver, just to have a look at what this data looks like. I'm specifying habitat and station as a factor here. It's important to understand the types of variables you have in your data frame because they're gonna perform differently with different analyses. So I'm just going to set these two as a factor, call it ggplot. So this is what our data looks like. You can imagine when I first saw this, I wasn't thrilled. But this is really a very bare minimum approach to range testing. You could do this a variety of different ways. You know, we basically spent like less than an hour at each site. We did it under near ideal conditions. So the idea was that we were trying to figure out what the maximum range is. So I would encourage a little bit more extensive range testing. You could do it off the side of the boat like we did, just placing a range testing transmitter, or you could station a couple of different reference tags for a little bit longer period too. It's good to have a general sense, though, of what your range is to even know where to put those reference transmitters in the first place. Because you could put them all too far away or all too close and not get the right information that you want out of this. So we're going to model this relationship. A lot of the literature will use logistic regression to model it. For me, I tried a whole bunch of different ones, and it might be the fact that this data is just a little bit sparse, but it seemed like a third order polynomial with a set y-intercept at 100 was the best model that explained the most variance in this data. And we're going to use that to predict the maximum range in the midpoint. We haven't done too much yet, is everybody? Everyone's still good? Yeah. So in order to do that, I'm not sure, I don't think we've worked with lists at all in this workshop, have we, Rob?
Yeah, so for the network stuff yesterday, I split the data frame by ID with the per map function instead of LPLS.
Okay. So first thing we're going to do is just set the intercept at 100. We're going to assume that right beside the transmitter you can always hear 100% of the time, which I think is a fair assumption. And then instead of fitting the model to every single receiver station, Sentinel station, like manually and fitting 9 different models, we're just going to split the data into a list and then apply the model using lapply to every receiver station in that list. And it's going to enable us to just do it very quickly and a lot less coding. And so first thing we'll do is make this df list thing using split by station. So each station is going to become its own object in this list. You can see that now. We have a list of 9 things, each of our stations, and the associated data with it. We're then going to apply that third order polynomial with the set intercept. So the -1, this is the model here, linear model. We're going to, detection efficiency is going to be our response variable. We're going to get rid of the intercept. we're going to fit a polynomial, third order, and then we're going to reset the intercept at the intercept value, which is 100. Whoops. So you now have this result coming out in the models. Again, it's a list, so it's not that useful for us just yet.
Jake, could you just quickly go over again how the LFY works?
Yep.
I think it might be.
So lapply is going to run over a list and apply the same set of operations to everything in that list.
The alternative would be rewriting the model just with a different data frame every time.
Yeah, so you could subset your data out and fit this model 9 different times if you wanted to too. But you could imagine, if you had an even bigger array and you had like 20 or 30 sites, lists can be very handy very quickly. Yeah?
Hey, I'm sorry if you mentioned this, but you mentioned reference times. They don't use code basic types to do the same kind of assessment like within a VPS. So you can look like components across a VPS with different environments or like time of day.
I think the VPS is sort of in its own functioning with having those reference transmitters in it are giving you very similar values to this DRC metric that you could model over space and time in the same way. Yeah. So we're just going to format the result. There's some other handy little things in here. So basically what we're going to do is run over that list again, and we're going to bind together the outputs where we're going to calculate from each model the coefficients, the R² value, the adjusted R², and the AIC value, which I assume you're all familiar with. So we now have this object that's drres. We'll just make it a data frame. We'll specify the site as the row names, and we can have a look at that. So we now have a data frame that has each of our Sentinel sites in it, and some of the information about the models that we fit to that relationship between distance and detection efficiency. And you can see we're modeling pretty good at most sites. This one we're explaining almost all of the variance. The adjusted R² compared to the R², by the way, is just accounting for the number of variables in the model. So we can make some predictions as well. So we'll generate a new data frame to make some predictions onto. So we'll just use this sequence function to generate a new data frame from 0 to 1,000 meters by 0.1. Set the intercept because the model needs to know the intercept to make predictions onto. And we're going to use that same lapply framework to make predictions out of our model. Bind those predictions back into our original data or distance generated data set. Just have a look at that. So we now have predictions for each site across distance. It's not in a very useful format though. This is in a wide format, and in order to plot it and do things with it, we want it in a long format. You can easily do this in dplyr. This is just sort of an I use reshape to do it, but this melt function is just sort of an artifact of I learned how to do this before I was using dplyr. So we'll make this new object. We need to tell it what our measured variables are, which are variables 3 to 11. Where they're going to call dplyr and rename our variables to something useful to us. And so we now have that exact same data frame that was in a wide format, just in a long one. We use ggplot now to plot that out. And this is something that I think we're all pretty familiar with at this point. So this is basically that plot from the paper that I showed you in the presentation earlier. Is everybody following along all right? No errors? All right. So we can there's also some useful code here. You're going to want to pull out from that model the predicted values for the maximum range and the midpoint, because you're going to want to use those two things later. Yep.
I'm just wondering for the predictions,
Yeah, I use the depth of the receiver. And for the most part, there wasn't any major differences in our system that way. The placement of that reference tag is really important, especially if you're just going to use one. There's potential for biases to creep in there if you're not picking a spot in the detection range of that receiver that's representative of the general detection range. So for example, if you're sitting on a slope and you put it up on the top of the slope, it might hear the tag very well, but it might hear down off the slope very poorly. And so the variation that's occurring in that detection efficiency of that reference tag doesn't represent the actual variance that's occurring in that area. And so if you have a really complex system, you might consider using more reference tags or put a lot of careful thought into where you're going to put it that would be representative of the overall range. And again, if you have more time and resources, I would certainly encourage using more reference tags. Yep.
Should you account for like animal ecology when you have, in terms of like the depth of that reference tag, like if you have a benthic animal, would you just put that tag perfectly at depth opposed to like maybe multiple because you've never really defined it?
Exactly. Yeah. That's the ecology of the animal is another very important consideration. Yeah. So we're just going to pull out the maximum range and the midpoint from this model. And we already have those predictions. So we're just going to pull them. We're just going to filter out the values that are 50 and 5%. Use a little if-else statement to just tell it what value this is. If it's 5, it's going to be maximum range. Otherwise, it's going to be midpoint. And so we now have this data frame that gives us for each one of our stations the maximum range and the midpoint. If you just want those in their own data frame, you can do that. So we'll generate midpoint in MR, and you can have a look at those. So these are our midpoint distances. These are our maximum range distances. So you can see in our system, because we're dealing with such a shallow, noisy environment, we're really not getting fantastic range. This one only hears 95, 96 meters in the maximum range. So we've gone out, we did our range testing, downloaded our receivers, ran it through this code, and so now we have this predictions of the midpoint. And so we can go back out and put our reference transmitters at this distance. And you see the distance is different for every single Sentinel site. It's very hard to get exactly 77 meters from a receiver, by the way. And that might be a part of the reason why those means don't come out perfectly like you'd like. And you might need to play around with it a little bit, test it, see how it's doing. Detection efficiency, generally speaking, also goes down over time, especially in places where receivers become biofouled.
Jake, if you did your test to obtain those data moving out one of those channels, let's say, with your boat, then would you have noted that that's the distance you would put your 78-meter tag? I mean, like it's not necessarily a homogeneous radius.
Yeah. Sorry, can you repeat the question?
So where you did your testing, like backing away with the boat,
Oh, with that linear line.
generate your curve. Now you've come up with your distances.
Mm-hmm.
Do you remember? Oh, we went down the channel, or we went up on the flat. Or
Yeah, that's basically what I did.
Put it there.
Yeah. Yeah.
GPS kept tracks and waypoints where you were testing, and you just go back to the reference tank there.
Yeah, that's a great idea.
Try to remember which way you're facing at each site.
Yeah, exactly. Yeah.
Yeah, because what's happening between 78 meters and 96 to drop 45%.
Yeah, it's crazy, isn't it?
Yeah. What's there? I want to know what's there.
Yeah. It's largely the fact that the further the transmission has to travel through the water, the less power it has
Yeah.
once it reaches the receiver. So and
Bathymetry, I'm
Yeah.
imagining, like, at that point, is that where your slope comes up and now you don't have
in my case, in my system, I have pretty good straight lines. It's not a physical obstruction issue. It's an environmental noise issue, I think.
Okay.
And so environmental noise with increasing distance from a receiver has an increasingly high impact on its ability to hear the tag,
And that's
just because of the power issue
Yeah.
with distance. So the same amount of noise, it just can't hear us. You know, it's at some distance the noise overtakes it, essentially.
Overtakes it, yeah.
So that's what's happening.
So that's what you're dealing with then.
I think so.
With this, yeah.
Yeah. Yeah. So this is just that same plot, but we're just going to add the midpoint and the maximum range to it. So this is that exact same plot from the paper and that we looked at before in the presentation. Again, I'd encourage doing a little bit more range testing than we did. But if you're really strapped for time and resources, this is kind of a quick and dirty way of figuring out what your range is. Code's working for everybody? So we got that range stuff out of the way. We've now gone out, placed our reference transmitters at each one of our Sentinel sites. It worked out perfectly. You got exactly mean, 50%. Fantastic. So you load that data in. And again, all these data sets are processed to some extent that I'm giving you. And so was Roberts, for example, yesterday. And there is a lot of fantastic resources out there from taking your telemetry data from the raw format into a more usable format like this. And OTN has a lot of those resources, a lot of that code on their website. So we have this reference tag data here. I'm just looking at the structure there. You can also just look at the top of it. So for every station and every study hour, we now have information integrated on depth, habitat type, tide height, tide state, benthos type, rugosity, and then the number of detections that we got from that reference transmitter. And you can see even in the top few values, it's wildly variable.
What's rugosity?
The amount of variability in elevation on the bottom. Snorkeling level with the substrate, just a general estimate. So I think I scored it between 1 and 3. 1 being like totally flat and 3 being like huge coral heads in massive seagrass banks.
So for generalizing that for future applications, how would you suggest integrating rugosity into a model where someone was building for a different environment? Is scoring at 1 to 3 a good standard that everybody should follow? You think, or
Probably not. I don't know. I'm sure there's like people who have more expertise on geography and things would come up with better estimates of slope and things.
Take a chain, measure the length of the chain, then drop it on the rocks, and then measure the difference between the chain fully stretched and then lay it out.
It's fantastic, yeah. Yeah, she suggested that you take a chain and lay it over the bottom and just measure the distance between that the chain stretches from the top down to the bottom. That would give you some estimate of the height. I suppose you could use like one of those long measuring tapes too. Something more extensive than my general estimates. So when you look at the structure of the data, we need to, we're going to model it with the random forests. I think that's what we're doing. So you want to make sure your data is in the right format. And so by running this structure operation on the data frame, you can look at the different types of variables in your data frame. And so a lot of these things are factors or numeric. You'll notice rugosity is 1, 2, 3. I decided at some point I don't want to treat it as an integer. I want to treat it as a factor for modeling. So I just changed that over here. This is another really important thing about analyzing telemetry data is to understand the difference between presence-only data and presence-absence data. So when you take in your data frame and you have all of these study hours, that's only when you actually have detections and data. So if you want to know information about truly animal presence-absence over time, you're going to have to generate a new object that is every study hour that you're interested in. summarize your detection data in some way and then bring that over into a different data frame. And then all of the NAs that come out in the new rows, those are rows that you don't have data for, so you can make those zeros. So we already know how to work with date time from previous workshops in R. So we're going to quickly make this date time object so we can work with time. Can't remember why I made this other ref data2 object. We're just going to work with that. Strift time's handy because you can use it to convert your date times to different formats. So in this case, we're just interested, we're going to aggregate our data by hour. So you can use strift time to take that date time POSIX object and change the format down to just hour. And then we'll make that a POSIX CT object again. And if you run str on it, you can see we now have hour, which just has the hour level information on it. And then we also have date time, which would have more date time information. So we're now going to use that hour object in this sequence function again to generate a new data frame called date time, where hour in that data frame ranges from the minimum of our data set to the maximum of our data set, and it's going to sequence out by hour. And you look at that, it's already a POSIX CT object. That's fantastic. We now have a data frame that is our whole time frame of our study. And so we need to repeat that time frame for every one of our stations. So we're going to make a new object called stations that's just calling any unique values out of our data frame. And then we're going to use the merge function to merge the datetime object with the stations object. And you can see if you click on it here, we now have every hour for every station combination. So in order to get our actual reference tag data, the number of detections combined over into that data frame, the first thing we're going to use is dplyr again, this group by function. We're going to group by hour and station, and we're going to summarize just the number of detections that occurred in each hour and station combination. You look at that, perfect. This is again a presence only data set, right? So that's why we generated that presence absence data set. So what we're going to do is we're going to make this new object where we're going to merge together that date time station, which is the presence absence data set, with the presence only data set. And there's some other things you can specify with this merge function, and you can see one of those here. All x is true, meaning x is the first object, y is the second object. So we want to make sure that it keeps every level of that presence absence data set as it merges the new one. It merges these two things together. And we want to make sure that as it merges, it's going to match the appropriate study hour and the appropriate station. So we now have this ref data 4 object. Click on the right one. There should be more NAs in this. That work right? I have to come back to that, I'm not sure. So basically, theoretically, you should get a lot of NAs coming out of this. Anywhere you didn't have data in the actual data frame should come out as NAs in the rows. And so this is just a simple little operation to say, let's go inside of this new object. Anytime detections is an NA, we're going to make it a 0. Ah, yes, of course. So I'd actually already summarized the data at this point. So this is just sort of an example of how you would do this. But all the detections are going to come out as 1 in this case because I had summarized the data previously. It's a little bit confusing, I'm sorry about that. So there's a question earlier about our tag delay with our reference tags. Unlike the range tags that are pinging really quickly, every 7 seconds plus a 3-second burst interval, so we expect them every 10 seconds with the range tag. But with the reference tags, they're pinging a lot less often and with some variability. And the reason that for this is because we're trying to avoid the potential for code collisions. And so if you had hypothetically multiple tags in the same area that were pinging at a very high rate and at a very fixed interval, they could hypothetically sync up and be blocking each other's detections every time. So that's why Vemco puts in these random tag delays. So ours is between 270 and 330. Got to add 3 seconds to that because of the burst interval on the tag. And so we would expect between 11 and 13 detections per hour as the maximum expected number. And so we'll assume conservatively, we can have a look at that using this histogram. Quite a few zeros. Zero inflated data sets are always fun to deal with. So we'll assume that 11 or more detections, because of that random delay, we'll assume anything from 11 to 13 is considered to be 100% of detection efficiency. So we're just going to calculate that out, calculate detection efficiency, assuming 11 is the max. You're going to get some numbers that are above 100% because there's going to be some 12s and 13s in there, so we're just going to make those 100. And if you look at that data set now, there's actually a lot more hundreds than there are zeros. If you look at that, the relationship, the histogram of this, over all of the different Sentinel site, you can see there's a lot of different relationships and a lot of different variable distributions in the data. And so basically what detection efficiency variance is doing is it's calculating a mean, and then it's figuring out how the variance in detection efficiency is occurring over time. There might be a better way to do this other than figuring out the difference from the mean, especially with these types of weird and variable distribution patterns. So I was going to bounce back and forth between the PowerPoint and R for a bit here, but I think that'll be too challenging with the video. So as you recall in that sort of concept diagram, we now have this detection efficiency variance value for every study hour at every one of our Sentinel sites. And so we can calculate the difference between that and the mean value at each Sentinel site to figure out the variance that's occurring from its mean. So the first thing we're going to do is calculate means. Where they're going to back assign those mean values into our original data set and then calculate the difference between the actual detection efficiency at the time and the overall mean value for that station or that Sentinel site. I also included some code in here if your mean it gets very far off from 50 and you think it might be affecting your data. I came up with this standardization approach where you can scale your data back into that plus or minus 50% if you'd like. So and this is all outlined in that paper. So DEVC is just it's a different variation of that DEV value. So in our data set, we now have detection efficiency of the hour, the mean for that receiver Sentinel type, and the variance that's occurring. So in this particular study hour, this receiver was hearing 32% lower than its average. Again, you have a lot of zeros and a lot of hundreds. Does that make sense to everyone? So you recall we made that maximum range object a while ago based on the range testing data. And so we're now going to take we're going to combine over into it that distance value from that maximum range. So we're basically assigning over maximum range into our data set. And so ref data now has not only the variance at the hourly level, but also that static measure of maximum range that came from the original range testing protocol. And then as I outlined in that equation on that one slide, this is that equation just operationalized in R. So you got the maximum range plus the maximum range multiplied times this correction factor, integrating the variance in detection efficiency at the time. And so this, you now have that DRC value. And so this value is what you could take and apply in any kind of modeling framework that you want. You know, you could be a random effect in a mixed effects model, or it could be an error term in a state space model or something. You see the minimum value. So you take that really low range site to begin with, that 95 meter detection range site, and then at a very, very poor detecting time period, it's now down to 47.5. Again, this is sort of a unitless measure, but it might be sort of analogous to meters. If you want to look at the relationship between that maximum range, the detection efficiency variance, and the derived detection range correction factor, I know there's a lot of terms here, I'm sorry. So you can get a sense about how, you know, that's sort of a similar looking relationship to what we saw in that hypothetical data set, right? Where you have the correction factor being driven primarily by that maximum range, but also variance occurring around that due to the detection efficiency variance of the reference tag at the time. This is kind of constrained a little bit in the sense that this correction factor can never really get super large or super small. So like if a hurricane comes through your study site, like Hurricane Irma did in 2017, probably detectability is 0 for a while. But with this method, it's never going to correct down to that level. And so there might be more to be done there to make this a little bit more, to expand the applicability of this to a wider range of conditions. Again, this is just something that we kind of dreamed up as a starting point, and I'm really hoping that people start to keep working on this and evolving it and making it more flexible and useful. 25 minutes. So we have this DRC value, but it's only at our Sentinel sites. And that's been very handy for us because we didn't have to measure detection range at every single one of we have over 100 receivers in the Florida Keys. We only measured at 9 of them. And so we're going to fit that random forest model to the data and then use that model to predict over our entire array. Or in this case, a subset of the different sites. So we have this ref data object. It's got all the variables that we need to do it. It's got that DRC value. It's got all the site-specific information as well as variability over time. And there's, again, getting all this stuff together can be tricky, and there is a lot of good R code out there on the OTN website to get from the raw data into this format. So again, rugosity, I already made it a factor before, but this is still in here. This is the time period before you do any modeling where you really want to look at the structure of your data and decide how you want to treat all your different variables. Make sure factors are factors and numbers are numbers when you want them to be. So we'll call random forest. And random forest, there is some randomness in them, as the name would suggest. And so you need to, in order to not have a different output come out every time you run the model, you want to set a seed. It's just a random starting point for the model. And when the model starts at that random starting point, it's going to give you the exact same output every time. And so it's reproducible for your papers. So you don't want to fit a model, put your data out there in your code, and have people going, well, I'm not getting the same output. This isn't reproducible. And so that's kind of an important part of that whole reproducibility and credibility thing in science. And the other great thing, to go back to that whole collaborative data analysis thing, is if you are doing collaborative data analysis working in GitHub, pushing and pulling your code and data in and out of the cloud, at some point, if you really want your data to be accessible and reproducible, you can just make that GitHub public at some point, or make a new public one with some, a cleaner version of your analysis. And that way, everybody knows exactly what you did with your data, can get reproducible examples. And when I look through the literature, I see those papers that do that getting cited a lot more than papers that don't. because they're reproducible examples and people understand them and they trust them. Yeah, Rob?
Sort of a random question I could have asked you privately, but maybe other people wonder as well. How do you choose what number for your set C? I always set this.
12 and 10 are my two favorite numbers.
Why not just 12?
That's the only reason.
Your bank's insured?
That's my social insurance number. Yeah.
The longer the more digits, the less efficient though. Why not just set it at 1 every time?
Yeah, I don't have a lot of expertise on this. It's
I have the exact same answer, so at least we're on the same page.
yeah.
Good.
I like 12 and 10.
Yeah. I always set 65.
So this is sort of the basic structure of how you can fit a random forest. They're pretty plug and play kind of a model. There's a lot that you can do with them, and there are some broader considerations, and I'd encourage you, if you're going to use them, to read into some of the caveats and benefits and some of the different examples. But this is just an example of a really simple one. So the first thing we're going to do is just define a formula. And I like to do this with all of my modeling exercises because, so Z is going to become a formula that is the model structure. So we're going to model detection efficiency, detection range correction factor, DRC, by habitat, benthos, rugosity, depth, diel, tide state, and tide meters. And by defining this formula, you can go through and fit all sorts of different models and just call the formula instead of having sort of clogging up your code with the, typing that formula at every time for every model. And so, when I landed on this model, I tried a bunch of other ones too, right? I just included the random forest, but the formula is nice and handy for that reason. We're going to make this object called a ref forest. Using the random forest, this is the random forest package. There's lots of extensions and different things you can do with this. Calling our formula, tell it what our data is. We're going to sample without replacement. And there's a lot to these. There's a reason why I chose that in terms of assessing variable importance. If there are any NAs, there shouldn't be. We're just going to omit them, but there's different things you can do. You can permutate them if you want to. We're going to calculate variable importance for our predictors, and we're going to fit 1,000 different trees. And so what it's doing is it's taking the data, randomly grabbing a set of data and a set of predictor variables, and fitting a decision tree to it. And so it's creating binary partitions in the data over and over again, trying to optimize the prediction of the response. And so it'll take habitat. It'll try to split up different habitats and see how well it can explain the variance in the DRC based on splitting at different habitat levels. And then different combinations, different habitats. We're going to split by these habitats and then by these depths. And it just keeps doing that randomly over and over again until it fits 1,000 different trees or however many trees you specify. And then it uses bagging to aggregate all the trees together to optimize the best performing model, the best predictions of your response. And so pretty much like all machine learning, its focus is really on prediction accuracy. And again, if you go onto my GitHub page, there's a workshop on applying these types of models to ecological data. So we'll fit the model. Zed's not found. It's going to take half a second to fit. What's that? Oh yes, of course. What do I like about random forests over boosted regression trees or support vector machines? The random forest is a very nice, well-developed set of tools around it for assessing variable importance and making predictions. You can also do really cool stuff with cross-validation. So you can do 10-fold K cross-validation in your data, where you're splitting your data into 10 different sets, training based on 9 of them, predicting onto the other one. And you do that over and over again, and you can partition your data out to deal with like spatial autocorrelation or temporal autocorrelation explicitly in the random subsetting process, which is really important for things like telemetry data sometimes. So there's a lot of utility with random forest models. I find the structure of the splittings to be very intuitive. It's something that people can look at the trees and understand very quickly. And the boosted regression trees are fantastic too, and they can, in some cases, be tuned to make better predictions than random forests do because of the way that they fit the data. But there's a lot more tuning and things to know about boosted regression trees to get them to fit well. So random forest is a little more plug and play than boosted regression trees are. So as I mentioned, the first thing that's going to come out when you fit a model is the percent variance not explained in the out-of-bag predictions, which is 24%. So if you just run print on that object, which is the model, ref forest, gives you 1,000 trees. It only tried 2 variables at each split. You can change that if you want. It's going to give you the mean square of the residuals and the percent variance explained. So we're explaining 76% of the variance in DRC based on this set of predictors. And you could, of course, include any ones that you're interested in. Wind might be a really important one as well that I didn't include. So you now have this model that seems to be fairly accurate, at least 76% accurate, at predicting DRC. And the important thing to note about this as well is this is percent variance explained in non-training data. And so integral to the modeling process of random forests, because it's randomly grabbing data and variables, it's able to assess its prediction accuracy in non-training data inherently. And so most modeling processes, what you want to do is split your data up like that k-fold thing that I was talking about before. Because you don't want to overtrain your model on the data. It might be explaining patterns that are specific to that data set and aren't generalizable patterns in the system. And so you really need to test your model on non-training data. And there's lots of different ways to do that. And random forest is nice because it does it inherently.
Question back here, Jake?
Yeah.
I forget if you said this, but how did you pick those variables? Did you run any testing to see which were most important?
The predictors?
Yeah.
Sorry, I'm going to forget that every time. I was asked, how did I pick those predictor variables? These were ones that, it's a combination of things that I thought would probably affect detection efficiency, as well as what I was interested in the actual ecology of the animal. So I was interested to know whether permit were using different habitats across different tide states. So naturally I wanted to put tide state into my model so I can make predictions at that ecological scale that I wanted to look at the data at. And there's lots of different procedures. These random forests are actually really great for variable selection. And so you can throw, 50 variables at a random forest if you want. And there's different extensions of it that are better at variable selection stuff. There's some tricks to it, but you can use random forest as a tool to to select your most important predictors and then use those in like a logistic regression if you wanted to. If you wanted a more traditional statistical framework where you have p-values, if you don't know which variables to use, random forest is a good one to figure out which your important variables are. And I think I didn't include this here. Yeah, this is just the importance values. And there's also some native plotting functions for these important values. But again, this is that same table I showed you where we have the different, and the way that it's assessing this is basically because it's randomly selecting variables and data, it's figuring out the amount of error that's incurred in the model by dropping that variable from the model. So each variable isn't used every time it fits a tree. And so it can figure out how important that variable is by seeing the difference between how well the trees perform when that variable is in the data versus when it's not in the data. $15 minutes. Yeah. So the question is, are the invariable importance scores as high good? Yes, high is better. High is a more important predictor. Because more error is incurred in the model by it not being in there. These are two different metrics. Mean squared error is overall performance accuracy, prediction accuracy. Node purity is how much it's contributing to the individual splits. So it's more related to the actual operation of the model. Everyone's blue? Believe how blue and green everyone stayed this whole time. The last thing we're going to do really quickly is pull in some permit detection data. This is just a very small subset of my data. Don't publish it on me. Everyone seems to be scared of that for some reason. Again, in order to make predictions with the model, you need to make sure you have your variables in your prediction data set. So we have this permit detections data frame that has all of the same variables in it as the model, or else the model's not going to be able to predict. It needs all those predictor variables in the new test set, and they need to be in the same format. So regosity needs to be changed over to a factor, or else the predictions won't work. We'll just use predict. We'll use predict based on the ref forest, that random forest model, onto permit detections. So we now have DRC predicted across this permit detections data set. We're gonna summarize it by site just so we can plot it out and see what's going on in the system. ggmap. So if you want to be able to pull satellite images from Google, it used to be very easy. Now you have to register an API with them. And so you go online, it's going to scare you because it's going to want your credit card information, and you think it's going to charge you, but it doesn't. So I get a bill every month from Google for $0, and I pull maps off of from them all the time through R. So what you have to do is go get that key, register an API, and you put that key in here, and you'll be able to pull maps yourself. What I did was I just included that map as an RDS file in this workshop. These are particularly useful for me because I work in shallow coastal systems, and the satellite images are really very telling of what's going on. So we're going to use ggmap, sort of like an extension of ggplot, and we're just going to plot the mean detection range values that we predicted onto our data set. Okay, similar to that plot that I showed you that's in that paper. If you want to animate it spatiotemporally, we can do that too. We just need to make sure that we have an hourly.
Why don't you use MarMap, Jake?
If I was on my computer, I would show you what MarMap looks like in the area. It's all the same depth. The entire area is, because it's all less than 4 meters. And so like basically the reef drops off and you get interesting depth contours out there, and then the entire area of the Florida Keys and Gulf of Mexico is just one flat area.
Really?
Yeah.
I've had terrible times dealing with the Google API code to get ggmap working.
Oh really?
Yeah.
Okay. Well, we could have a look at it and see if we can get it working.
I'm down.
Okay. Offer rescinded.
I appreciate it.
So again, we're going to make an hourly variable here that we're going to plot across. We're going to lon transform our detections just to plot them. And we're going to subset it for just the first couple of weeks because otherwise it's going to take us forever to animate this thing. So we're going to call ggAnimate. We've already been introduced to this before. Make this P object. I don't know why I always call my plots P. And the only real difference between this and the ggmap is that we now have this transition time, which is that hour POSIX object. And we're also going to add a title. That's the time frame. Never seen that before. So we have 166. Ooh, why is this weird? We have 166 different hours, and so we're going to animate this with 166 different frames, so that every hour has its own frame. We're going to make the video 20 seconds, or the animation 20 seconds. And this is probably going to take about a minute to produce because it's literally making this same plot 166 times. And then it's just going to play it over in a loop. This is one of those fun things to have computer races with. See who is the fastest computer.
This is really good.
Yeah. Well, it's nice that pretty much all the packages have already been used too for other workshops.
Like, and everybody walks away with a computer that has all these tools on it. So it's
Yeah.
pretty awesome.
That's sweet.
We're going to get with Dave here. He's got a weird NAN kind of error.
Oh.
And it seems like he's doing the same things everybody else. So I'm going to dig into the error results a little bit, but I'm pretty confused. The rest of the folks I might bring back, but
Okay. Huh. Man, are so frustrating like that?
I feel like you could take this as far as you want it. You could take this so far. Doing all your range testing,
Oh yeah.
like you could spend your entire season doing range testing. Take this further, starting adding slopes and all these other variables. Anything you possibly can.
you got a bigger study area and you can run a slope come around with a tag on it. You can also have a bunch of like on-board, real-time, like matching environmental conditions you're matching at.
Oh yeah.
You could have that. You could have real time.
Yeah.
We did some stuff in Halifax line, but we haven't tried like doing this yet, like actually going back and applying where we did do the range testing to the rest of the line. But this is like a pretty great framework for this getting to that and doing it. Yeah. Excited to try to convince some of the field guys.
Yeah, I mean, it seems really common where people do the range testing but don't really know exactly what to do with it, or just report it in the paper, like, hey, this is my range.
That's what you see all.
Yeah. It's like, we did range testing, check that box, right? But I don't know, it's really hard because,
Yeah.
there's so much to it, and as you said, you could spend forever
And
doing
when
it.
do you stop?
Yeah,
Yeah. When do you stop?
yeah. When's it good enough? You're never going to actually quantify that like crazy three-dimensional envelope that is your detection range, right?
Yeah.
So it's like some reasonable measure
Yeah.
that you can use.
And like you said, you can see your outliers. Like it'll show you, okay, this channel is only getting 70. Yeah. And this is
So this is just kind of a fun animation. I don't know if you guys have got this to run yet, but it's just showing the variation of that DRC value over time throughout that whole study period. And then permit detections are kind of just flashing in and out of the different sites. This is more for fun, not so much for real science, but so we got about 5 minutes left. The last little bit of this code is just applying that correction factor to the number of permit detections in the system. It's pretty simple. We don't really need to run through it too much. It's going to produce those exact same plots that we saw in the presentation and are in the paper. Just run right through. Huh. Interesting. Anyone else get that error? Stat summary. Anyways, again, as I said before, I wouldn't necessarily recommend this application in terms of just correcting your number of detections. I think that DRC value has a lot better applications in terms of modeling stuff. But all the tools are here to, if you go out the field and collect that data and you've got all this R code to run it, you should be able to work through it, I think, pretty reasonably. And so, that's the method. I hope you guys find it useful. I hope you continue to work on it and develop it. Appreciate your feedback. And again, there's lots of great code on this. If you go to my GitHub page, you go to the OTN page, there's lots of good stuff for these types of analyses. So thank you all for your attention.
Testing, testing. Looks like we're good.
You don't have to.
Okay, thank you. All right.
I just do not want to see myself on the screen. Anything else I can do?
All right, we're good. Thank you very much, Jake, for that talk. We're going to switch directions now, and we're going to head over. I got to stop walking in front of that light. Okay, we're going to head over. We're going to take a break from coding and start to talk about grant writing, okay? So Joanna Mills-Fleming is here today. She's actually my PhD supervisor, so she and I have been working together for a fairly long time now. And she's also been a part of OTN since its conception. So she's been a really big part of the data committee, coming up with new statistical methods for animal tracking data and new visualization methods as well. She's also a member of the scientific advisory committee of OTN. Her personal research interests include developing statistical methodologies for spatial and temporally correlated data. And so she doesn't just do animal movement modeling, but she's also really big and had a lot of success in the fisheries industry as well. And so she's, luckily for me, said that she would help us out and give us a little talk on the success of her grant writing and how her tips for actually conducting grants. And we're lucky too because she's also been a part of the NSERC Discovery Grant Review Council for 3 years. And so thank you so much, Joanna.
My pleasure. I'll just get hooked up here. Thank you, Kim. I hear you guys are doing an awesome job. So I'm disappointed I can't actually participate more. So as Kim said, is that too close or are we okay? Okay. As Kim said, I'm a statistician and a full professor in the Math and Stats department here at Dell. I've been here sort of since the get-go. So I started off as an assistant professor and moved through the ranks. And I've been asked to talk to you about grant writing. So I thought in order to keep your attention or convince you to listen to me, I'd tell you a bit about sort of how I've gained this experience, if you will. So I'm currently incoming chair of the Math and Stats Liaison Committee for NSERC. So the job of that committee is really to help NSERC guide their awards program, so the Discovery Grant Program, all the way up to how they allocate funding to the Institute. So I realize you're not all mathematicians and statisticians, and so may not be familiar with some of these, but BERS, PIMS are big sort of institutes here in Canada that fund research in mathematics, statistics, more recently data science. And this committee sort of guides how NSERC should spend their money to run those institutes. I'm also chair of the Statistical Society of Canada Research Committee, which does more or less the same thing, but is the voice for basically statisticians in Canada. This may be useful, especially to those of you who are sort of early career researchers, so students, postdocs. I am a member of the scientific review panel for the postdoctoral program for ERMS, which is again sort of mathematics geared, but a lot of applications for postdocs in some sense, as hopefully I'll convince you of here in a few minutes, are somewhat generic. Perhaps my biggest or greatest experience I've had in terms of both grant writing and reviewing comes from chairing the NSERC 1508 Discovery Grant Evaluation Group. And so essentially what that afforded me the opportunity to do was to read over 300 discovery grants. And the last year I served on that committee, I was the chair, so I was really responsible for ensuring that those grants were given out fairly across this country. In addition to sort of the 300 or so statistics applications, I also have reviewed nearly 100 applications that went to 1503, which is ecology and evolution. So if you're in Canada and you're doing quantitative work, which obviously some of you are, then it could be 1503 that you apply for a discovery grant. And so that was really a tremendous amount of work. Reading, as many of you know who've applied or prepared even, say, just one grant, it's a huge amount of time to prepare these, and it's also a huge amount of time to review them and make sure that they're reviewed fairly. And I think if I wish actually now, I was thinking about this morning, that I'd actually had somebody talk to me early on in my career about how to prepare a grant. And I'm not saying that to sort of slam Dalhousie. I think perhaps this sort of opportunity is far too often, and maybe it's something you sort of put on the back burner and you think is sort of too generic in terms of how it's presented by the Faculty of Grad Studies. Who knows? But there's a few sort of hints and tips that I have for you that I do truly believe would have helped me to secure more funding and earlier on in my career. And so hopefully what I've told you in the last few minutes gives me some credibility to do just that. I should also tell you that I've been successful in obtaining discovery grants, and in the last competition I got an accelerator, which is essentially sort of a big pot of money on top of the usual amount, which allows you to go on and do more exciting things a lot faster. And so that's sort of been a notable thing in my career, I would say. I think I should also add that after coming back from NSERC, so essentially when you serve on this committee, you go up and you spend a week in Ottawa, and about 4 months prior to that on the phone every week discussing grants, or applications for grants, I should say. So I came back and I was pretty enthusiastic about how much I'd actually learned. At the time, my PhD student, Ethan Lawler, had decided to apply for a Vanier. And so I mean, Ethan has a Vanier because Ethan is a fabulous student, but I felt like for the first time in my career, I could guide him through that application process with much more sort of certainty and perhaps confidence than I had before. And he actually won a Vanier. He was the first statistician to be awarded a Vanier in this country. So I attribute a little bit of the service I did to NSERC to that success. So all that said, here are some of my tips. And I know there's some people from the US in the audience who will have to apply to NSF instead of NSERC. NSERC is moving. It's not moving quickly, but in terms of the number of successful applications that are funded every year, it's going lower. So it's getting more competitive, and it's harder to get an NSERC Discovery Grant than it ever has been before. So in the US, the number's around 20%. In Canada, it's been upwards of 80%, and it is going down fast. So there is no room in the system, and there is no point in putting in a rushed application. I have done it. I was on maternity leave. I had like a 3-month-old baby. I wasn't getting enough sleep. I said, oh, I should just do it because then it's done. There is no room for that sort of application in this system anymore. So if you don't think you can prepare the grant properly within the time frame that you have, try and defer it to the year following, because it's in some sense a wasted effort if you don't. And I don't think you can apply every year. So having now being in the situation where I can look back and say, why did I rush that? Really, for the sake of the year and doing something properly, it's worth it, because even if you're not successful, you'll get much more useful feedback. So I guess part and parcel to that is to start early with these applications. Keep your CCV up to date, because that's just a pain. The CCV system is actually changing once again, but if you keep all of your important information, so your papers, what's in progress, what's in submission, what's in review, then that saves a lot of time at the last minute pulling things together. So I actually sometimes follow my own advice. I tend to keep my CCV up to date for that very reason. The unfortunate but fortunate thing is also, especially for the Discovery Grant competition, the rules are changing all the time. So how you would prepare a grant last year is going to be slightly different than how you'll prepare it this year. Case in point, The last year I was serving on the committee as chair, we were told by NSERC that there was going to be a small but significant component devoted to EDI. Okay, so if in the year that rolled out after you had a comment saying that you thought seriously about EDI, it was noted. The year after that, it was necessary. So if you can pay attention to those rules, you can even get sort of the language in early. You need to think deeply about things like equity, diversity, inclusion, and how you're going to handle those in your labs and in your workplaces. And it was very interesting to me how diverse, in fact, the approaches were, but how tremendously valued they were by the evaluation group. So when new things come in like that, they're totally, in some sense, unrelated to your actual raw research, but they're essential to your application. So if they're looking for a statement on EDI, make sure it's in there. And make sure that it somehow links into your program. This one sounds like the sort of thing I say to my 2 teenagers: follow the directions. And I'm not sure, perhaps it's just how I went through school, and in university I was very science-based. But when you read a description of what's supposed to be contained in your application, it needs to be there. So things like short-term objectives, long-term objectives, nobody has the time. Each application is reviewed for 15 minutes. It's discussed for 15 minutes. No one has the time to go through and sort out what's a long-term objective and what's a short-term objective. They have to be staring people in the face. So if you're asked for something like that, make it obvious. That sounds trivial, it sounds like it shouldn't really be a value, but it's essential because of how the reviewing process works. So be meticulous, and I feel like I would have had a great deal more success early on. I think my research program has been generally well thought out, but I don't think it was as well described as it could have been had I paid attention to those bullets. So do what you're asked. Download the grid. So I actually brought the grid. It's tricky to find, and it will obviously be different if you're getting funding through NSF or something, but there is a grid that has to be made public by NSERC that changes every year, that tells the evaluation group how to rank or how to assess the applications. It talks about the criteria for the merit of the proposal, the excellence of the researcher, and the training of HQP, which is highly qualified personnel. You can go on and you can see exactly what people are looking for. And so having a look at the grid, the last time I applied to NSERC, I looked at that grid and I could guess what my score was going to be. Not because they were all one side of the grid or the other, but because I knew what was in my application. And so if you've written a good application, you should be able to look at that grid and have a sense of where you're going to fall on it when your application is reviewed. Another, and this again pertains specifically to the Canadian system, write a good notification of intent. That is critical. Far too often I hear people say, okay, I just got to do that over the weekend. The notification of intent actually is what is used by the committee to determine who will review your application. Okay, it's not the whole proposal. By the time we get the proposals in January, the committee has already been assigned. So if you think that your application, for example, justifies joint review, so you want an ecologist and you want a statistician or a data scientist, that needs to be conveyed in the NOI because it will not be changed after, or very rarely is it changed. So the NOI and doing a good job of that is almost as essential as writing the research proposal. And that's a thing I think that goes missed more often than not. You will have every right to look at the membership. So you can, I actually went on to this morning just to make sure I had this right. The Discovery Grant competition was last week. You can go on and look at who's on the Discovery Grant committee for your particular group. And you can tailor your NOI. If you know a person or you look at their research and you decide, well, that aligns well with what I do, they're going to appreciate this, then think about the language that you use in the NOI and how you can attract them to reading it. Okay, that is your right and privilege. Don't worry too much about the external reviewers. So you are at liberty to propose reviewers. I was shocked at how little impact they have on the application assessment. And there's a few reasons for that. It's not that NSERC is looking to waste people's time. The main reason is that often those reviews are not done or the instructions aren't followed. So when I ask someone to review a grant application for me, I send them the instructions. I highlight the parts that I think are most critical. Because what a reviewer can't use the grid. A reviewer can say, wow, this is a great proposal. But unless they give reasons, unless they follow or answer the questions asked of the reviewer, then it doesn't get much weight at all. So I guess the main point here is if you're going to list reviewers, select people you know are going to take the job seriously, do it on time, and highlight the parts that you see as most critical to the success of your application and make them aware of that. I've told you it's 15 minutes per application. That number shocked me to start with. You spend months preparing something like that, and in often less than 15 minutes it's done. Make things easy to find. Make all the things that are asked of you available and clear in the proposal. The other thing, and nobody told me this, I will tell you, the first time I applied to NSERC, I didn't actually mention that I'd been on maternity leave for a year. Because I thought back in 2005 that would be seen as lacking energy and enthusiasm or something. That is a mistake. So the way NSERC works, so we are tasked with evaluating you within a 5-year window or 6-year window. I think it's 6 now. If you were on, say, paternity leave for 6 months of that window, the committee is required to evaluate you in a 5.5-year window. Okay, so the window adjusts. You are not doing yourself a disservice if you are on leave or if you are ill. That should be made clear in the application. Obviously not the details, but your interpretation of how it affected your window. So if you were off sick for 3 months, say you were off sick 3 months and it took you another month maybe to ramp back up. It has to be in there. You can't leave the committee, so you can't say, hey, I was ill during the window. You have to make your case. I was ill for X months, and this is how many months in the window it affected. The committee then has all the information that they need, and the onus is on you to report that accurately. But report it. It's really essential. I've seen great applications where it's obvious that there's been something happen, and if it's not stated, we can't infer. So make that information crystal clear. So I said that a different way here on my notes. Tell your story. If you're on sick leave or any sort of leave, make it clear. State how it impacted your window. Okay, and look at the rules. There's all sorts of rules outlined by NSERC which tell you what is appropriate and what is not. It's all there. And this is not meant to scare you. It is getting more and more competitive, and it's getting more and more competitive very, very quickly. Stats, for example, in the competition that ended last week had a record number of applications, which is good and bad. If you get rejected, it is not the end of the world. You can walk around with your head down, hating NSERC, saying that it wasn't done fairly, and it gets you nowhere quickly. It is both prestigious and I actually believe essential in this country to hold an NSERC grant. And so you have to figure it out. The best way is to look at the information. So if an application is rejected, the onus is on the evaluation group to communicate clearly why it was rejected. And in my experience, that's done well. So you will get a collection of information back, which indicates what was missing and why. And you include that in the next go-round. You can also reach out to NSERC. There are program officers that will then dig out the application and go right through it with you from start to finish. You lift your head up and on you go. And you'll get one next year. So there are going to be more and more people that are rejected. And it's just that there's not enough money to go round. And remember that the committee itself is not trying to determine if an application is funded or not funded. The committee scores the application. Then completely independent of that, the program and group leaders decide of the money allocated to this particular group, how many bins are we going to fund? So you get put in a bin depending on your score. And it may be that one year bin J, which is the bin you're in, gets funded, and one year it doesn't because there just wasn't enough money left. And that is basically life. So it sucks, but it is the way it works. And I truly believe, having seen all of these different, I've seen applications right from ECRs, so first time out, early career researchers, no training of HQP, all the way up to, what they call outstanding on all three criteria. These would be leaders, people who are doing groundbreaking work that's impacting a very broad community, and I really believe there's a place for everyone. So you have to work hard, but making sure that you tell your story is essential. So there's a whole bunch. When I first started writing discovery grants, I was really focused on the research proposal, making sure it was innovative and original. And that is a huge part of it, but there's other parts that if you don't have in will just completely mean that you miss the mark because it's so competitive. A few more specifics, and this is really for the Canadian system. Get involved in the training of HQP. So NSERC calls students. They could be undergrads, masters, PhD, postdocs. Postdocs maybe is a bit of a stretch if you're still a student yourself, but get involved. It is from NSERC's perspective, it is no more valuable to be training a PhD student than it is to be doing an undergraduate honor student. They are valuable contributions to the training of HQP. So get involved. Nobody counts up, this person had 3 PhD students and this person had 2. It doesn't work that way. You have to show that you've done quality training. Question? So you evidence that by telling your story. So if you were, say you were on, you were a supervisory committee for an honors project, write it in your application. There's a section devoted to the training of HQP. And I think people get caught up in thinking, oh my gosh, I've only been involved with 1 undergraduate student and no master students. You have to give evidence that you've done something. So get involved now. And I truly believe it doesn't necessarily, it's not a numbers game. Just like papers. It's not all about how many sole author papers. It doesn't matter as long as you make sure that your contribution is original and innovative. It doesn't matter if you're a co-supervisor of a PhD student or a sole supervisor, as long as your role is clear. So some of the metrics that I think, especially for somebody my age, were sort of brought up to think are essential are not essential. What's essential is making sure that the work you're doing is quality, it's innovative, it impacts the community. The broader the community it impacts, the better off you are. A lot of it's to do with impact. And you also have to make the case for what impact means for the research you do. Is it industry impact, government impact, academic impact? Who knows? explain your contributions. You don't need 17 single author papers to get an NSERC grant. Okay? Demonstrate that the research is quality, impactful, important to a broad community. Sorry, Kim, I've got 2 more things. And I guess finally, and this is important, especially on the dark days of writing grants, NSERC does not fund projects. NSERC funds programs. So all the parts, the training of the students, your EDI plan, all of them need to link. And that's what makes these time-consuming to write. So you want to have a program. You want to have short-term objectives, long-term objectives, even with so far as impact. You want to explain what your career aspirations are. So I'll stop there. But hopefully that gives you a sense of what NSERC is looking for, and as a result, the academic community here in Canada.
We have some time for questions for sure, if anybody has some.
I'm sure I could look this up, but you're here. I'm just curious, how much would weighting between your CV versus the quality of your proposal be?
So they're equal weight. So there's 3 criteria, not 2. So there's excellence of researcher. So that's going to really come into your publications, your output. The next is the merit of the proposal. So you could be off the charts or off the grid with the merit of the proposal, and perhaps mediocre on excellence of research. Or maybe you've been out in industry, you haven't been doing research. But you can really kill it with the merit of the proposal if you write a good proposal. And then the third part is the training of HQP. And that's where it gets very tricky for young people, which is why I'm saying get involved, informally or formally. Because NSERC does allow for early career researchers to get a moderate on HQP, which is generally not fundable, but is for ECR. So there's a bit of a catch-all for that. But notwithstanding that, if you've done a little, then it takes you sort of out of the moderate and into the truly fundable, which just means a higher bin and more money.
Just a follow-up question on the training. How do workshops like this look compared to supervision? I mean, they're both important, or?
They're both important, and really, and again, it's up to you. Make the case.
Yeah.
Say how many people. So say this is your discovery grant application. Tell them how many people were here. All of that sort of thing. The other thing that is useful or essential if you have had trained students is where do they go? So they don't actually have to go, to Stanford and get an assistant professorship to have impact. It could be that they go work in the line of research I do, they go work for Fisheries and Oceans Canada. Who knows? But make it clear that it's an impactful position if it is.
Yeah, following up on that again. So, for example, I do a lot of student mentoring, but a lot of the time it's not in an official capacity, like I'm not on a committee. Basically, my interpretation of what you're saying is that I need to start to be getting on more official roles.
Just quickly, could you just repeat the question too?
Yes, sorry. So the question was, I do a lot of I would call it informal supervision of students. And the comment was, it sounds like you're telling me to sort of formalize some of that. I would formalize it. And it just means that you devote less of your space to trying to make a case. If you can say, I'm on these 4 committees, ask to be on the committees. I've never turned anyone down who wanted to be on a students committee because it's added value. So ask to be on those committees and formalize it wherever you can because it just makes it easier. And you're not, it's the same amount of work.
Any other questions?
If you get rejected for a discovery grant, what are the other avenues you can go down to maintain a research program or develop new avenues that can build up a CV and work towards getting a discovery grant?
So I think I could speak to those. So Yeah, so the question is, if you do get rejected from the Discovery Grant system, what else can you do? So my hope is that most people going off into academia, in this country at least, are given a startup grant. And part of the reason, so I still have some of my startup grant. It was some tiny little amount in 2005, but I still have it. And it's kind of emergency money. So you can pretty much survive for a bit on a startup grant, and I would negotiate that when I take a job and get it to be as big as you can for exactly this reason. I think I can only really speak to what other outlets are there in the math and stats world. So CANSI, which is the Canadian Statistical Sciences Institute, has a variety of programs. I would search out people on campus that do work like you do and look to big granting opportunities. So Sarah Iverson here was one of the people who got me involved in the OTN. And that has been tremendously impactful to my career and a lot of fun. Being part of these bigger grants is energizing and fun, and you shouldn't do one and not the other, but often those are helpful if you do have a year where you don't have Discovery Grant funding. So I would get involved in bigger grants too. And I would use all of the things I've said, even when you're involved in those bigger grants. It doesn't come for free, so you're still asked to comment and help with the application itself and read them and really add value based on this sort of thing, because so many people are working so hard that sometimes you miss some of the sort of easy things to tick off.
So look for lower hanging fruit. Another question I had, you mentioned that if you are involved with student mentorship, I think you said for an early career researcher, a moderate
Is okay.
mentorship is okay. So are there different bins for early career researchers and other researchers? And what are the bins and how are they defined?
Oh, these are good questions. Okay, so the way it works, so the question is, you mentioned moderate on a bin. What does that mean? Okay, so do I have 2 minutes?
Yeah, of course. If you have 2 minutes.
Or I have 2 minutes. So basically, I brought this just to show you, this is the grid that was used in 2017 when I was first a member of this committee. And I was scared. I was in Ottawa. I was like, how do I make sure I'm doing this properly? So you spend the first night calibrating. So we have a whole bunch of mathematicians, applied mathematicians, and statisticians in one room. And we're all very different. Okay? So basically, the way it worked was we were all supposed to make sure that for any given proposal, we would, in these 3 sort of subgroups, we would evaluate people the same. So essentially what happened is you're scored on these 3 criteria. I've said them once already, but they're important. Excellence of researcher, merit of proposal, and training of HQP. So those are the columns, if you will. Across the top goes from insufficient to moderate, to strong, to very strong, to outstanding, to exceptional. I don't mean to scare you, but really you need to be strong, strong, strong. Generally, based on how the funding envelope looks in Canada, you need to be over here to get funded. Everything in orange is the tiny language differences. So listen to this. So this is EOR, Excellence of Researcher. To get a moderate, the accomplishments presented in the application were deemed to be of reasonable quality, impact, or importance. That is not going to get you funded. The accomplishments presented in the application were deemed to be solid in their quality, impact, and or importance. That's a strong. That is fundable. Exceptional is the highest category. Acknowledged as a leader who has continued to make, over the last 6 years, influential accomplishments at the highest level of quality, impact, and or importance to a broad community. So when you first look at this grid, you're like, how do I do this? But I will tell you, most people are somewhere in the middle. There's very few out here, and fortunately there's very few in the insufficient tail. Insufficient, if you don't have short and long-term objectives, you go right back there. That is where, so there's a lot of research programs that if you don't follow the NSERC guidelines, you can still end up down there. If you have an incomplete CCV, all of these things which I've seen people, and they call it the halo effect in NSERC. So you will see somebody who has done amazing work for years, who decides, I don't have time to complete my CCV, and it's not going to matter because I'm that good. Well, it matters, because you can get an insufficient if we can't see what we need to see to evaluate it properly. So there's all sorts of things that make it so that you can be successful on this grid, but you need to know what's expected of you, which is why I say read the instructions. Because there's no rolling or flipping a coin here. It really means you just have to put in what's asked for.
Any other questions? Okay, let's thank
Okay.
Joanna again.
Does it make you want to read?
Okay, thank you again so much, Joanna. That was incredibly informative. I really asked Joanna to come here today, and I asked her for a specific talk about NSERC because she has so much experience in it, and because it is what a lot of us, we've been targeting this whole, we've targeted this workshop to early career researchers, which means you guys will likely be applying for these, and that's why I really wanted her here today. But we also have some international researchers who won't be applying for Canadian grants, and so although the information was very specific, but I think it really applies to a lot of different granting procedures. And so just thank you tremendously. That was absolutely incredible. For the next hour, we will be on lunch, and I'm going to put up the GitHub page again. Please try and download YAPS and TMB and install them on your computer before we start the afternoon lunch session, because we have two fairly complicated modeling techniques that are coming up, and it would be really nice if we could avoid any technical difficulties right from the get-go, okay? With that, enjoy your lunch. Thank you.